Gene therapy is a direct application of genomics where faulty genes are replaced, corrected, or silenced to treat diseases. The use of CRISPR-Cas9, a genome editing tool, has made it possible to precisely modify DNA sequences. Clinical trials are ongoing for CRISPR-based therapies for diseases like sickle cell anemia, beta thalassemia, and Leber's congenital amaurosis. In vivo and ex vivo strategies are being optimized for safety and efficacy. Genomics also plays a vital role in vaccine development, particularly in the design of recombinant and mRNA vaccines. For instance, the mRNA-based COVID-19 vaccines, Pfizer-BioNTech and Moderna, were developed using genomic information from a SARS-CoV-2 spike protein. Reverse vaccinology, which involves mining pathogen genomes for antigenic proteins, has led to vaccines against Neisseria meningitidis and holds promise for others like malaria and HIV. In public health, genomic data informs population screening programs, epidemiological studies, and policymaking. Projects like the 1000 Genomes Project and the All of Us Research Program collect diverse genomic data to understand how genetic diversity impacts health outcomes and to ensure Projects like the 1000 Genomes Project and the All of Us Research Program collect diverse genomic data to understand how genetic diversity impacts health outcomes and to ensure equitable healthcare. Projects like the 1000 Genomes Project and the All of Us Research Program collect diverse genomic data to understand how genetic diversity impacts health outcomes and to ensure equitable healthcare. However, the application of genomics to health also raises ethical, legal, and social issues such as privacy of genetic data, informed consent, and potential discrimination based on genetic information. These concerns are addressed by frameworks like GINA, Genetic Information Non-Discrimination Act, and guidelines from the Human Genome Organization, HUGO. In summary, genomics is reshaping the landscape of healthcare by facilitating precision diagnostics, individualized therapy, early disease prediction, and advanced vaccine development. Its integration with technologies like artificial intelligence, big data analytics, and mobile health platforms holds immense potential for transforming global health, making medicine more predictive, preventive, and personalized. Its integration with technologies like artificial intelligence, big data analytics, and mobile health platforms holds immense potential for transforming global health, making medicine more predictive, preventive, and personalized. Previous year CSIR-NET questions with answers. CSIR-NET Deck 2016. Which of the following techniques is most appropriate for identifying genetic variants associated with complex human diseases such as diabetes and asthma? APCR. B-gel electrophoresis C genome-wide association study GWAS ddNA cloning. Answer: C genome-wide association study GWAS. CSIR-NET June 2019. A patient with breast cancer shows HER2 gene amplification. Which of the following therapeutic agents is most appropriate in this case? A. Imatinib. B. Trastuzumab C. Tamoxifen D. Paclitaxel. Answer: B. Trastuzumab. CSIR-NET Deck 2022. Which of the following is used as a genome editing tool in gene therapy? A. Southern blotting. B. ELISA. CCRISPR-Cas9 ddNA microarray. Answer: CCRISPR-Cas9. Let me know the next topic from Applied Biology, and I'll prepare it in the same high-quality format. You said, agriculture, ChatGPT said. Here is your CSIR-NET JRF Life Sciences lecture on Applied Biology, Agriculture. Maximize detail, JRF level, based on previous year question trends, fully paragraph-based, no tables, with 2 to 3 previous year questions and answers included. Agriculture, as a branch of applied biology, integrates principles of plant science, genetics, biotechnology, microbiology, soil biology, and ecology to enhance crop productivity, resistance, and sustainability. The modern approach to agriculture emphasizes understanding biological processes at the molecular and organismal levels to improve crop yields, reduce losses, combat climate challenges, and ensure food security. The area is highly relevant to the CSIR-NET syllabus under Applied Biology, where focus areas include plant breeding, pest and disease management, biotechnology in agriculture, and sustainable farming practices. The Green Revolution, which began in the 1960s, marked the first major application of biological knowledge in agriculture. It involved the use of high-yielding varieties (HYVs) of crops, chemical fertilizers, and irrigation systems. Semidwarf varieties of wheat and rice, developed through conventional breeding, drastically increased productivity. However, this also led to environmental issues such as soil degradation, water depletion, and pesticide overuse. Consequently, modern agricultural biology has shifted towards sustainable practices, molecular breeding, and genetically engineered crops to reduce environmental impact and increase resilience to stress. One of the significant breakthroughs in agricultural biology is plant genetic engineering. Genetic modification enables the introduction of desirable traits such as pest resistance, herbicide tolerance, drought resistance, and enhanced nutritional content. The introduction of Bt crops e.g., Bt cotton, Bt brinjal, where the cry gene from Bacillus thuringiensis is inserted into the plant genome, imparts resistance against Lepidopteran pests by producing insecticidal proteins that disrupt the gut of the insect. Bt cotton has been widely adopted in India and has led to reduced pesticide use and increased yields. Herbicide-tolerant crops are another innovation, such as Roundup Ready soybean and maize, engineered to tolerate glyphosate. These allow farmers to control weeds without damaging the main crop. However, overuse has raised concerns about herbicide-resistant weed evolution and biodiversity loss. Molecular breeding and marker-assisted selection (MAS) have improved the efficiency of conventional plant breeding. Molecular markers linked to disease resistance or stress tolerance can be used to select for desired traits at the seedling stage, accelerating the breeding process. For instance, resistance genes like Exa21 for bacterial blight in rice and LR34 leaf rust in wheat have been successfully deployed using MAS. CRISPR-Cas9 and other gene editing technologies now allow for precise editing of endogenous plant genes without the introduction of foreign DNA. For example, editing of the sweet gene family in rice has provided resistance to Xanthomonas infections. Since these edits may not involve transgenes, they can potentially bypass strict GMO regulations, opening new doors for crop improvement. Soil biology and microbiome management are also key areas in agriculture. Understanding the soil microbial community helps in enhancing nutrient uptake and plant growth. Biofertilizers, including nitrogen-fixing bacteria like Rhizobium, Azotobacter, and Frankia, and phosphate-solubilizing microbes, promote sustainable nutrient cycling. Mycorrhizal fungi, especially arbuscular mycorrhizae, improve phosphorus uptake and drought tolerance. Biopesticides, derived from natural organisms like Trichoderma, Bovaria, and Nucleopolyhedrovirus, are gaining popularity due to their specificity and environmental safety. These are used for managing plant pathogens, nematodes, and insect pests in eco-friendly ways. Plant tissue culture techniques, such as micropropagation, somatic embryogenesis, and anther culture, are widely used for mass production of disease-free leaf plantlets, conservation of rare species, and generation of homozygous lines in breeding programs. Clonal propagation of crops like banana, sugarcane, and orchid through tissue culture has revolutionized horticulture and floriculture industries. Climate-resilient agriculture involves breeding crops tolerant to abiotic stresses such as drought, salinity, flooding, and heat. DREB, HKT, and NHX gene families are known to confer stress tolerance. Transgenic crops with overexpression of such genes are under development and testing. Nutritional improvement through biotechnology is exemplified by golden rice, which contains genes from daffodil and bacteria to produce beta-carotene, a precursor of vitamin A. This biofortification approach is being applied to several staple crops to combat micronutrient deficiencies. Integrated pest management (IPM) combines biological, cultural, physical, and chemical tools to minimize pest damage. It includes crop rotation, natural predator conservation, resistant cultivars, and judicious pesticide use. IPM reduces environmental impact and delays pest resistance development. Sustainable agriculture emphasizes conservation of biodiversity, minimal chemical input, and ecological balance. Practices include organic farming, use of compost and green manure, crop diversification, and agroforestry. In conclusion, applied biology in agriculture has evolved from traditional selection to precision genome editing and ecosystem-based management. The convergence of molecular tools, bioinformatics, and ecological knowledge is redefining crop improvement and agricultural sustainability. Understanding these concepts at both fundamental and applied levels is crucial for CSIR-NET aspirants aiming to master this area and solve real-world problems in agriculture and food security. Previous year CSIR-NET questions with answers. CSIR-NET June 2017. Bt cotton plants are genetically modified to express which of the following to resist insect attack? A EPSPS enzyme. B. Cry protein gene from Bacillus thuringiensis. C. Glyphosate resistance gene. D. Virus coat protein gene. Answer: B. Cry protein gene from Bacillus thuringiensis. CSIR-NET Deck 2021. Golden rice is genetically engineered to be rich in iron. B. Zinc. C. Vitamin A, beta-carotene. D. Vitamin D. Answer: C. Vitamin A, beta-carotene. CSIR-NET June 2018. Marker-assisted selection is best described as a hybridization-based crop improvement. B. Selection based on yield alone. C. Using molecular markers linked to desired traits for breeding. D. Physical selection based on plant morphology. Answer: C. Using molecular markers linked to desired traits for breeding. Let me know the next topic you want from applied biology or any other CSIR-NET unit. I'll prepare it in the same format. You said. Genomics and its application to agriculture. ChatGPT said. Here is your CSIR-NET JRF Life Sciences lecture on genomics and its application to agriculture. Max word limit, exam-oriented, JRF level, no tables, no wasted space, with 2 to 3 previous year questions and answers. Genomics, the study of an organism's complete set of DNA including all its genes, has revolutionized modern agriculture by providing precise tools for crop improvement, stress resistance, and yield enhancement. In agricultural biology, genomics is not merely a tool for gene identification but forms the foundation for molecular breeding, transgenic development, disease diagnostics, and sustainable farming practices. The advent of high-throughput sequencing technologies, bioinformatics, and functional genomics has allowed scientists to explore gene function, expression profiles, and regulatory networks in plants at an unprecedented scale. The primary goal of agricultural genomics is to understand how genes control agronomically important traits such as drought resistance, flowering time, disease resistance, nutrient use efficiency, and grain quality. Through whole genome sequencing, WGS, genomes of important crops like rice, Oryza sativa, maize, Z-maize, wheat, Triticum estivum, and tomato, Solanum lycopersicum, have been fully sequenced. These reference genomes allow for the identification of genes and QTLs, quantitative trait loci, associated with complex traits. One of the earliest milestones in agricultural genomics was the International Rice Genome Sequencing Project, IRGSP, which provided a complete and annotated rice genome, enabling researchers to link gene functions with stress responses and productivity. Similarly, genomic information in maize and wheat has facilitated the mapping of genes involved in kernel development, disease resistance, and abiotic stress tolerance. Comparative genomics between model plants like Arabidopsis thaliana and crops allows identification of conserved genes and regulatory pathways. For example, the flowering locust TFT gene, first studied in Arabidopsis, plays a central role in flowering regulation and has been targeted in crops to alter flowering time and photoperiod sensitivity. Functional genomics tools such as transcriptomics, RNA-seq proteomics, and metabolomics provide insight into gene expression patterns under stress conditions or developmental stages. These datasets help identify candidate genes that can be used for trait improvement via either marker-assisted selection, MAS, or transgenic methods. RNA-seq data have helped reveal how rice and wheat respond at the gene level to drought, salinity, and pathogen infection. Molecular markers, including SSRs, symbol sequence repeats, SNPs, single nucleotide polymorphisms, and AFLPs, are derived from genomic information and used extensively in marker-assisted breeding. For example, SNP markers linked to the sub-1 gene in rice, which confers submergence tolerance, have been used in breeding flood-tolerant varieties like Swarna sub-1. This allows integration of traits without affecting the rest of the genome, making breeding faster and more precise. Genome-wide association studies (GWAS) use natural population diversity to associate specific genetic loci with traits, offering higher resolution than traditional QTL mapping. GWAS has been used to identify genes related to nutrient uptake, such as nitrogen and phosphorus efficiency, pest resistance e.g., against stem borers and flowering time in many crops. A transformative development in genomics is the use of CRISPR-Cas9 genome editing in agriculture. Unlike traditional GMOs that introduce foreign DNA, CRISPR allows targeted mutation or replacement of endogenous genes. In rice, CRISPR has been used to knock out the BADH2 gene, enhancing aroma. In tomatoes, editing RIN genes has improved shelf life. In maize, Argos8 gene editing conferred drought tolerance. These gene edits are often indistinguishable from natural mutations and are being explored for regulatory approval as non-GMO. Pangenomics, which involves sequencing multiple genotypes of a species, provides insight into genetic diversity, presence-absence variation, and rare alleles linked to traits. In crops like maize, where genome size and complexity are high, pangenome studies help in utilizing diverse germplasm for breeding resilient varieties. Transcriptome profiling during abiotic and biotic stress helps identify stress-responsive genes. For example, under drought stress, genes encoding late embryogenesis abundant, leak proteins, dehydrins, and DREB transcription factors are highly expressed. These stress-related genes can be used to develop transgenic or gene-edited crops. In pathogen resistance, genomics has enabled the identification of resistance R genes and defense signaling pathways. Resistance genes like Pi54 in rice against blast disease, RPG1 in barley against stem rust, and year 15 in wheat yellow rust have been characterized and used in breeding. Genomics-based diagnostics can also detect emerging pathogen strains and help track resistance breakdown. Genomic selection (GS) is a breeding approach where genome-wide markers are used to predict the breeding value of individuals. GS is especially useful for polygenic traits like yield and climate resilience. It reduces the breeding cycle and increases selection accuracy. Metagenomics, which analyzes DNA from environmental samples like soil, helps understand the plant microbiome and its role in nutrient uptake, disease suppression, and growth promotion. This knowledge is being used to develop microbial inoculants and biofertilizers. In precision agriculture, genomic tools assist in tailoring crop management practices to the genetic potential of a variety in the local environment. By integrating genomics with phenomics, high-throughput phenotyping (AI), and remote sensing, farmers can make informed decisions about planting, irrigation, and fertilization. Genomics-assisted biofortification targets genes involved in micronutrient accumulation. In rice and wheat, genes controlling iron, zinc, and vitamin A synthesis have been modified to combat malnutrition. In conclusion, genomics has emerged as the backbone of modern agricultural biology. Its integration into breeding, diagnostics, and crop improvement strategies enables a sustainable, productive, and resilient agricultural system. For CSIR NET aspirant, understanding the principles, tools, and real-world applications of genomics in agriculture is essential, as this area is frequently targeted in the exam and forms the basis for cutting-edge research in life sciences. Previous year CSIR NET questions with answers. CSIR NET June 2019. The sub-1 gene introduced into rice through marker-assisted back crossing confers tolerance to a drought. B salinity C submergence D cold stress answer, C submergence. CSIR NET Deck 2020. In crop genomics, the CRISPR SOS9 technique is used primarily for increasing genome size. B inserting random mutations C precise editing of specific genes D generating RNA interference constructs answer, C precise editing of specific genes. CSIR NET June 2022. Which of the following statements about genome-wide association studies GWAS is correct? A GWAS only uses SSR markers. BGWAS can identify loci associated with complex traits. CGWAS is less accurate than QTL mapping. BGWAS can only be used in animals. Answer: BGWAS can identify loci associated with complex traits. Let me know the next topic you want from Applied Biology or another unit of CSIR NET Life Sciences, and I'll continue in the same high detail, exam-oriented format. You said, gene therapy. ChatGPT said. Here is your CSIR NET JRF Life Sciences lecture on gene therapy. Maximize word limit, JRF level, paragraph format, no tables, based on previous year questions with answers in year mentioned. Gene therapy is a transformative approach in molecular medicine aimed at treating or preventing diseases by directly modifying the genetic material within a patient's cells. It represents one of the most advanced applications of molecular biology and biotechnology in clinical practice. In gene therapy, defective or missing genes are corrected, replaced, or silenced using various delivery methods. The goal is to address the root cause of genetic disorders, rather than merely alleviating symptoms. The concept of gene therapy rests on the central dogma of molecular biology, where a functional gene produces a functional RNA and protein. In many genetic disorders, a mutation disrupts this flow, leading to a defective or missing protein. Gene therapy seeks to restore this pathway by introducing the correct version of the gene or modifying the existing genetic sequence using tools like CRISPR, SOS9, zinc finger nucleases, ZFNs, or TALENs. These molecular scissors can precisely target and edit specific DNA sequences in vivo or ex vivo. Gene therapy can be classified into somatic gene therapy and germline gene therapy. In somatic gene therapy, the genetic alteration is restricted to somatic cells and is not heritable. It is the only form approved for clinical application in humans. Germline therapy, which involves modification of gametes or embryos, remains ethically controversial and is currently prohibited in most countries due to the risk of permanent heritable changes. Another classification distinguishes in vivo gene therapy, where the gene is delivered directly into the patient's body, and ex vivo gene therapy, where cells are genetically modified outside the body and then transplanted back. Ex vivo methods are commonly used in blood disorders like severe combined immunodeficiency, SCID, and beta thalassemia, as they allow precise control over gene delivery and screening of modified cells before reintroduction. Vectors are essential for gene delivery. Viral vectors such as retroviruses, lentiviruses, adenoviruses, and adeno-associated viruses, AAVs, are commonly used because of their high efficiency in gene transfer. Retroviruses integrate their genome into the host DNA, providing long-term expression but carry risks of insertional mutagenesis. AAVs are favored for their low immunogenicity and stable gene expression. Non-viral vectors, including liposomes, naked DNA, and nanoparticles, offer safer alternatives but usually have lower efficiency. Gene therapy can be classified into somatic gene therapy and germline gene therapy. In somatic gene therapy, the genetic alteration is restricted to somatic cells and is not heritable. It is the only form approved for clinical application in humans. Germline therapy, which involves modification of gametes or embryos, remains ethically controversial and is currently prohibited in most countries due to the risk of permanent heritable changes. Another classification distinguishes in vivo gene therapy, where the gene is delivered directly into the patient's body, and ex vivo gene therapy, where cells are genetically modified outside the body and then transplanted back. Ex vivo methods are commonly used in blood disorders like severe combined immunodeficiency, SCID, and beta thalassemia, as they allow precise control over gene delivery and screening of modified cells before reintroduction. Vectors are essential for gene delivery. Viral vectors such as retroviruses, lentiviruses, adenoviruses, and adeno-associated viruses, AAVs, are commonly used because of their high efficiency in gene transfer. Retroviruses integrate their genome into the host DNA, providing long-term expression but carry risks of insertional mutagenesis. AAVs are favored for their low immunogenicity and stable gene expression. Non-viral vectors, including liposomes, naked DNA, and nanoparticles, offer safer alternatives but usually have lower efficiency. Unmark in gene therapy was a successful treatment of X-linked SCID using a retroviral vector to introduce the IL-2RG gene into bone marrow cells. Despite initial success, some patients developed leukemia due to insertional activation of oncogenes, highlighting the need for safer vectors and precise integration control. The lentiviral vector system, with a safer integration profile, later emerged as a preferred alternative. The emergence of CRISPR-Cas9 technology has revolutionized gene therapy by allowing site-specific editing of the genome with unparalleled accuracy. This system uses a guide RNA to target a specific DNA sequence in the Cas9 nuclease to induce a double-stranded break. The break is then repaired by the cell's endogenous DNA repair machinery, leading to gene correction, disruption, or insertion. CRISPR is being tested in clinical trials for diseases such as sickle cell anemia, Leber congenital amaurosis, and beta thalassemia. In cancer therapy, gene therapy strategies include suicide gene therapy, where a gene encoding a toxic protein is delivered selectively to cancer cells, and immune gene therapy, which enhances the immune system's ability to recognize and attack tumors. One of the most successful examples is CAR T-cell therapy, where T-cells are genetically engineered to express chimeric antigen receptors (CARs) that target cancer antigens such as CD19 and leukemia and lymphoma. Gene therapy is also being explored for neurodegenerative diseases, such as Parkinson's disease, spinal muscular atrophy (SMA), and Huntington's disease. For instance, Zolgensma, an AAV-based gene therapy for SMA, delivers a functional copy of the SMN1 gene and has shown remarkable success in infants with this fatal disorder. Despite its promise, gene therapy poses several challenges. These include immune responses to vectors, transient expression, off-target effects especially in genome editing, and the difficulty of targeting specific tissues. Moreover, large-scale manufacturing, regulatory approval, and cost are significant barriers to widespread clinical use. From a regulatory standpoint, gene therapy trials are closely monitored by agencies like the FDA and EMA. Ethical considerations, particularly around germline editing, human enhancement, and long-term effects, are under active international debate. The first CRISPR baby case in China, 2018, triggered widespread condemnation and stricter regulatory oversight worldwide. In veterinary medicine, gene therapy techniques are being explored for improving livestock health, productivity, and resistance to disease, though the primary focus remains in human therapeutics for now. In conclusion, gene therapy represents the pinnacle of personalized molecular medicine. Its integration with genomics, bioinformatics, and synthetic biology promises a future where genetic diseases may be not only treatable but curable. For CSIR-NET aspirants, mastering the principles, techniques, and clinical examples of gene therapy is essential, as this area is conceptually rich, frequently tested, and at the frontier of biotechnology research. Previous year CSIR-NET questions with answers, CSIR-NET Deck 2018. Which of the following gene therapy methods is classified as ex vivo? A direct injection of viral vector into muscle. B. Delivery of genes via nanoparticles into lungs. C. Genetic modification of bone marrow cells outside the body and reinfusion. D. CRISPR-Cas9 based gene editing in brain tissue. Answer: C. Genetic modification of bone marrow cells outside the body and reinfusion. CSIR-NET June 2021. The risk of insertional mutagenesis is most closely associated with which of the following vectors used in gene therapy? A. Adenovirus. B. Retrovirus. C. Liposome-based vector. D. Naked DNA. Answer: B. Retrovirus. CSIR-NET Deck 2022. Which of the following statements about CRISPR-Cas9 is correct in the context of gene therapy? A. It introduces random mutations across the genome. B. It cleaves RNA in a sequence-specific manner. C. It induces site-specific double-stranded DNA breaks. A. It introduces random mutations across the genome. B. It cleaves RNA in a sequence-specific manner. C. It induces site-specific double-stranded DNA breaks. D. It always integrates foreign DNA into the host genome. Answer: C. It induces site-specific double-stranded DNA breaks. Let me know the next topic in applied biology or any other unit of CSIR-NET Life Sciences, and I'll continue in the same in-depth and exam-focused manner. You said. Bioresource and uses of biodiversity. ChatGPT said. Here is your CSIR-NET JRF Life Sciences lecture on bioresources and uses of biodiversity. Maximized word limit, JRF level, paragraph format, based on CSIR-NET Life Sciences previous year question areas, note tables, with 2 to 3 solved PYQs. Biodiversity, or biological diversity, encompasses the variety and variability of all forms of life on Earth, including genetic, species, and ecosystem diversity. Bioresources are the components of biodiversity that hold actual or potential utility to humans, such as plants, animals, microbes, and their products. The study and application of bioresources, a core area in applied biology, has profound implications for medicine, agriculture, biotechnology, and sustainable development. The Convention on Biological Diversity (CBD) defines bioresources as genetic resources, organisms or parts thereof, populations, or any other biotic component of ecosystems with actual or potential use or value for humanity. The uses of biodiversity can be broadly categorized into direct, indirect, and option future potential uses. Direct uses involve tangible biological products like food grains, vegetables, fruits, fish fiber, cotton, jute, coir, fuel, wood, dung medicines, quinine, artemisinin, taxol, and industrial products, rubber, resins, oils. Indirect uses relate to ecosystem services such as pollination, nutrient cycling, climate regulation, water purification, and soil formation. Biodiversity also underpins cultural, spiritual, and recreational benefits. Option values reflect the future, as yet unknown, uses of biodiversity, which may be critical for medical or technological innovations. Bioresources in agriculture include crop wild relatives and land races that carry genes for resistance to pests, diseases, or environmental stress. These are crucial in crop improvement programs via plant breeding or genetic engineering. For example, wild varieties of rice and wheat have contributed genes for drought and salinity tolerance in modern cultivars. Similarly, microbial bioresources are exploited for nitrogen fixation, e.g., rhizobium, azospirillum phosphate solubilization, Bacillus, Pseudomonas, and biopesticide development, Bacillus thuringiensis. In pharmaceutical sciences, over 25% of drugs used in modern medicine are derived from higher plants, many of which are from tropical rainforests. Examples include morphine from Papaver somniferum, aspirin from Salix alba, and vincristine from Catharanthus roseus. Additionally, marine bioresources, such as sponges, corals, and cyanobacteria, were rich sources of novel anti-cancer and anti-inflammatory compounds. Biodiversity also fuels biotechnological innovation. Enzymes from extremophiles like Thermus aquaticus, source of Taq polymerase used in PCR, or Pyrococcus furiosus, used in high-temperature industrial processes, illustrate how genetic diversity translates into technological breakthroughs. Metagenomics allows access to genetic resources of uncultivable microbes from diverse environments, enabling novel enzyme and metabolite discovery. Genetic resources are protected under international treaties like the Nagoya Protocol, which governs the access and fair benefit sharing of genetic material. The International Treaty on Plant Genetic Resources for Food and Agriculture (ITPGRFA) ensures farmers' rights and conservation of crop diversity. In India, which is one of the 17 megadiverse countries, bioresource management is governed by the Biological Diversity Act, 2002, implemented through the National Biodiversity Authority (NBA). This act ensures sustainable use, conservation, and equitable benefit sharing. Traditional knowledge associated with biodiversity is documented in the Traditional Knowledge Digital Library (TKDL) to prevent biopiracy, as in the famous case where the U.S. patent on turmeric's wound healing properties was revoked. A key concern is the erosion of biodiversity, driven by habitat destruction, pollution, invasive species, overexploitation, and climate change. Loss of biodiversity not only reduces ecological resilience but also deprives humanity of potential bioresources, which could otherwise address future health, food, and climate-related challenges. Biodiversity is also being economically evaluated using models like total economic value (TEV), which includes direct use, indirect use, and non-use values, existence and bequest values. Modern conservation approaches like bioprospecting, searching for valuable genetic and biochemical resources, community-based conservation, and ecosystem services valuation are integrating science with economics and ethics. In conclusion, bioresources are an integral part of biodiversity with immense actual and potential value. Their responsible utilization, equitable sharing, and long-term conservation are essential for human well-being, sustainable development, and ecological balance. For CSIR Net Life Sciences, this topic is critical because it integrates ecology, biotechnology, policy, and ethics, areas that are often cross-linked in multiple sections of the exam. Previous year CSIR Net questions with answers, CSIR Net June 2016. Which of the following is a correct example of indirect use of biodiversity? A. Medicinal plants. B. Firewood collection. C. Pollination by bees. D. Collection of wild fruits. Answer, C. Pollination by bees. Explanation. This is an ecosystem service, not a direct harvest of biodiversity. CSIR Net Deck 2019. The Indian law that governs access to biological resources and associated knowledge is Environment Protection Act, 1986. B. Forest Conservation Act, 1980. C. Biological Diversity Act, 2002. D. Wildlife Protection Act, 1972. Answer, C. Biological Diversity Act, 2002. CSIR Net June 2022. Which of the following is a correct statement regarding bioresources? A. They are restricted to genetically modified organisms. B. They include only plants used for agriculture. C. They include all biotic components with potential utility. D. They are limited to medicinal plants and microbes. Answer, C. They include all biotic components with potential utility. Let me know the next topic in applied biology or any unit from the CSIR Net Life Sciences syllabus. I'll continue. You said. Here is your CSIR Net JRF Life Sciences lecture on breeding in plants. Maximum word limit, JRF level, CSIR Net focused, previous year question based, paragraph format, no tables. Plant breeding is the art and science of improving the genetic makeup of plants to enhance desirable traits such as yield, quality, disease resistance, and stress tolerance. This discipline, foundational to agriculture and food security, uses principles of genetics, reproductive biology, molecular biology, and biotechnology. Traditional plant breeding relied on phenotypic selection and hybridization, while modern approaches incorporate marker-assisted selection, tissue culture, genetic engineering, and genomic tools. CSIR Net Life Sciences often tests conceptual understanding, breeding strategies, and integration of molecular tools with classical genetics in plant breeding. The process of plant breeding begins with germplasm collection and evaluation. Germplasm refers to the genetic material available for breeding, encompassing land races, wild relatives, traditional cultivars, and mutant lines. The variability present in germplasm is the foundation of breeding efforts. Following evaluation for desirable traits, breeders use selection, either mass selection selecting many plants with desired traits, or pure line selection selecting and propagating progeny from a single superior individual to improve populations. Hybridization is the crossing of genetically distinct parents to combine favorable alleles. The resulting F1 hybrids often exhibit heterosis or hybrid vigor, showing superior qualities like higher yield or stress resistance. However, in self-pollinated crops like wheat or rice, hybrid vigor tends to break down in subsequent generations, so selection must follow hybridization to fix desirable traits. For cross-pollinated crops e.g., maize, composite and synthetic varieties are developed through recurrent selection to maintain heterozygosity. Breeding for disease and pest resistance uses gene-for-gene interactions and relies on the identification of resistance R genes in wild relatives. Classical backcrossing introduces a resistance gene into a high-yielding cultivar, while retaining agronomic traits. In the Green Revolution, for instance, IR8 rice was developed through hybridization and selection for dwarf stature and high yield. Mutation breeding, induced by chemicals e.g., EMS or physical agents e.g., gamma rays creates novel alleles that are then selected for desirable traits. This has been successful in crops like groundnut and barley. However, the mutations are random and require extensive screening. The advent of molecular breeding revolutionized plant breeding. Marker-assisted selection (MAS) uses molecular markers RFLP, SSR, SNP linked to traits of interest to select desirable plants even in the absence of phenotype. This is particularly useful for traits that are difficult to screen phenotypically, such as root architecture or drought tolerance. The integration of QTL mapping allows breeders to identify genomic regions associated with quantitative traits, essential for complex trait breeding. Genomic selection (GS) is an advanced approach where genome-wide markers predict the breeding value of individuals, increasing selection accuracy and reducing breeding cycles. This is especially relevant in perennials or long gestation crops like coconut or oil palm. Transgenic breeding introduces foreign genes e.g., Bt, herbicide resistance into crops to improve resistance and quality. However, regulatory hurdles and biosafety concerns limit its widespread use. Genome editing, particularly using CRISPR, SOS9, is a frontier in plant breeding. It allows precise edits in endogenous genes to improve traits like flowering time, nutrient efficiency, and stress tolerance without introducing foreign DNA, making the plants non-transgenic in some regulatory contexts. Breeding also addresses abiotic stress resistance, an emerging priority in the face of climate change. Traits like drought tolerance, salt resistance, and temperature resilience are polygenic and complex. The identification of transcription factors e.g., DREB, NAC, and stress-inducible genes has facilitated molecular breeding for such traits. Plant breeders must also consider intellectual property rights (IPR) and farmers' rights, especially in countries like India under the Protection of Plant Varieties and Farmers' Rights (PPV and FR) Act. The balance between innovation and equity is crucial for sustainable breeding systems. In conclusion, plant breeding is a dynamic field integrating classical genetics, molecular tools, and biotechnological innovations. For CSIR-NET aspirants, understanding the methodologies, principles, and applications of plant breeding, particularly hybridization, marker-assisted selection, QTL mapping, mutation breeding, and genome editing, is essential. Questions often test your ability to connect genetics with applied breeding outcomes and understand trait inheritance under various breeding schemes. Previous year CSIR-NET questions with answers, CSIR-NET Deck 2016. Which of the following is an advantage of marker-assisted selection, MAS, in plant breeding? A. It allows direct visualization of desired traits. B. It eliminates the need for hybridization. C. It allows selection of plants based on molecular markers linked to traits. D. It increases heterozygosity in crops. Answer. C. It allows selection of plants based on molecular markers linked to traits. CSIR-NET June 2020. Backcross breeding is generally used to A. Generate heterozygous lines. B. Introduce a single gene into a superior variety. C. Create transgenic plants. D. Develop polyploid varieties. Answer. B. Introduce a single gene into a superior variety. CSIR-NET Deck 2017. In marker-assisted selection, the use of molecular markers is advantageous because they are always dominant markers. B. They eliminate the need for any phenotype evaluation. C. They can help in selecting traits not expressed early in development. D. They are only applicable in mutation breeding. Answer. C. They can help in selecting traits not expressed early in development. Let me know the next topic you'd like detailed. We can continue building your full applied biology coverage or move to another unit of the CSIR-NET Life Sciences syllabus. You said, and animals, ChatGPT said. Here is your CSIR-NET JRF Life Sciences. Animal breeding refers to the controlled propagation of domesticated animals to improve desirable traits such as milk production, meat quality, egg-laying capacity, disease resistance, fertility, and growth rate. It forms a critical pillar in animal husbandry and sustainable livestock development. In the context of CSIR-NET JRF Life Sciences, questions focus on classical and modern genetic principles applied to animal breeding, inheritance of quantitative traits, artificial selection strategies, and integration of molecular tools like marker-assisted selection and genomic selection. The foundation of animal breeding lies in population genetics and quantitative genetics. Traits of economic importance in animals are usually polygenic, influenced by multiple genes, and show continuous variation, making quantitative trait loci (QTL) mapping and heritability estimation essential components of animal breeding programs. Selection intensity and response to selection depend on the heritability of traits in the selection differential, as explained by the breeder's equation, R equals H squared times S, where R is the response, H squared is heritability, and S is selection differential. Inbreeding and outbreeding are two principal strategies in classical breeding. Inbreeding involves mating between related individuals to concentrate desirable alleles but increases the risk of inbreeding depression, where deleterious recessive alleles become homozygous. This is sometimes strategically used to fix desirable traits. In pure lines, such as in laboratory model organisms like mice. Outbreeding, including crossbreeding, outcrossing, and species hybridization, aims to enhance heterozygosity and exploit heterosis, hybrid vigor, leading to improved performance in F1 individuals. For example, breeding of Bos indicus, Indian breeds, with Bos taurus, exotic breeds, has enhanced milk productivity in cattle under Indian climatic conditions. Artificial insemination (AI) is a pivotal tool in modern animal breeding, allowing for genetic dissemination from elite males across large populations, even internationally. It reduces disease transmission and improves selection accuracy. AI forms the backbone of progeny testing, wherein the performance of offspring is evaluated to determine the genetic merit of breeding sires. This is a common practice in dairy cattle. Improvement programs. Open nucleus breeding systems (ONBS) represent structured breeding strategies where superior animals are maintained in a nucleus herd, and genetic gains are disseminated to commercial herds. This hierarchical model helps maintain genetic progress and control in large-scale programs. The integration of molecular markers in animal breeding has revolutionized selection accuracy. Marker-assisted selection (MAS) uses DNA markers, e.g., SNPs, microsatellites tightly linked to QTLs or major genes.